So continuing with the interdisciplinary research theme for the day, I would like to introduce Professor Harmit Malik, who actually graduated in chemical engineering from IIT Bombay. And Jitu, some of you may not know, he graduated in chemistry from IIT Bombay and then moved on to biosciences later. So Professor Harmit Malik is currently a professor and associate director at Fred Hutchinson Cancer Research Center and was recently inducted into National Academy of Sciences for his contributions in understanding evolutionary conflicts. And he's someone who's very passionate about mentoring and science communication as well. To those who follow his Twitter timeline, my interactions with him over Twitter have been mostly over memes, some of whose examples you will get to see today later during the break. With that, over to you, Harmit. Looking forward.
Thank you very much, Mohit. Can you guys all hear me okay?
Yes.
Okay, great. Fantastic. I'll just share my screen. I know that we are on pretty tight timelines, so I'll try to get to the meat of the matter right away. So thanks everyone for having me come and join the YIM meeting. This is actually my first YIM meeting. I have to apologize a little bit because my voice is a little affected by recovering illness. So I hope you can sustain through the rest of my talk as well. So I'm going to present my talk in two bits. I'm going to give you a little bit of science to get you started, and then I'll give you a little bit of my own sort of personal anecdotes and advice about navigating a career in academia, which requires a lot of skills, not all of which you actually learn during the course of your training. Work in my lab is actually focused on genetic conflicts, and the concept of genetic conflicts emerges from this fictional character, the Red Queen that you're all seeing on your screen, who says to Alice in the Alice in Wonderland books that in Wonderland, it takes all the running you can do to keep in the same place. And if you really need to get somewhere, you need to run at twice the speed. This was actually, in the context of the books, another example of how weird Wonderland is, but was seized upon as a very important theorem in evolutionary biology by Lee Van Valen and his colleagues. Actually, the Red Queen hypothesis was coined in 1973, which also happens to be the year I was born. And in a way, the simplest way to think about the Red Queen hypothesis, although there are a lot of nuances, is this is the second iteration of Darwinian thinking in evolutionary biology. Whereas Darwin was very interested in how organisms adapt by natural selection to their abiotic environments, Lee Van Valen and his colleagues recognized that a large part of that environment was made up of competing species. So for example, it was not possible to just look at these snow leopard populations in isolation without actually considering what was happening to their prey populations, which were the snow hares. And so if you were to actually map the population sizes over the years, you'll actually find these beautiful cyclical episodes of boom and bust cycles associated with these two species because their fitnesses are so directly interlinked with each other. In a way, we can extend this paradigm to study a lot of arms races in the molecular sense, and that's kind of what my lab does. For instance, it's very easy to extend this paradigm of a prey-predator relationship to that between antiviral proteins encoded by either the adaptive or the innate immune system and viral proteins that they're actually encountering. So in this cartoon example, the antiviral protein is going to recognize some feature of an incoming virus within the cell, and by virtue of that recognition is going to mount.
Harmit, you are on mute.
Sorry, can you hear me okay now?
Yes, yes. Yeah.
Okay, sorry about that. I don't know how much of that you missed.
Just half a minute.
Okay. I'll actually go back because it's nice to give a little bit of homage to the inspiration behind my work. This is sort of what I'm trying to do, which is we're sort of extending interaction between prey-predator relationships to that between host immune systems and antiviral and viral proteins that they're encountering. As we are all meeting on Zoom, here's another evidence of a worldwide effect of a viral infection that is in its works. But in a Darwinian sense, this is an engine for evolution because this is forcing the viral population, once it's recognized and antagonized by the antiviral protein, to be now adapting such that it's no longer recognized, and therefore the virus is temporarily winning this race. And that works until the immune system again reestablishes its dominance. If this is the adaptive immune system, antibodies and T cells, this can happen in the course of days. If this is your innate immune system, this can actually take years to decades to establish because these are all encoded or hardwired in the genome. Nonetheless, the fact that we are all here in spite of encountering many pathogenic viruses suggests that there are many lessons to be learned just by looking backwards in evolution in the sequences of the viruses that we've encountered and how the innate antiviral systems have adapted in response to this. And what drives this engine is the fact that one party is always losing this arms race. Therefore, there's always going to be an evolutionary advantage to be gained by innovation. What this means is that even though we sort of grow up in biology focusing on what is actually deeply conserved in evolution, in other words, we can look at this very simple metric of how amino acid altering nucleotide substitutions versus non-altering substitutions, so-called silent sites, accumulate in the course of evolution. So what you'd actually see if you were to do this analysis for all protein coding genes in your genome, this is sort of the dotted line references the speed limit that's actually provided by mutation, where if there was no selection acting, everybody would be clustered around a DNDS of 1. But you can see that most protein coding genes are far to the left. That suggests that on average, amino acid altering mutations are actually deleterious and they're purged from the population. So we call these slow evolving genes at the protein level. And really most of biology has focused on these genes because we focus on what is deeply conserved in evolution as a way to infer functions. But my lab is actually very interested in fast evolving genes, which are really indications where there is a lot of adaptation going on within the genome. And unsurprisingly, immunity genes are heavily overrepresented in this category. So what we've done is actually taken a retrospective view of rapid evolution to really identify specificity domains, premised on the very simple assumption that if this were a battle for binding affinity, whether the antiviral protein is going to avidly interact with the viral protein to mount its antiviral response, or the virus is going to escape, we actually expect the signature of rapid evolution not to be randomly distributed over these proteins, but actually to be concentrated on those residues or domains that maximally affect the binding affinity interactions. And this has actually turned out to be a very successful strategy to identify the specificity domains, even in the absence of any underlying biochemistry. What's really neat about this is it also tells us a lot about susceptibility determinants. And this is actually important, especially in today's context of having a pandemic raging, because these determinants can vary between species, but also can vary between different populations of the same species, which is our own. So just a very brief sort of précis of how we did this work. This was the very first example where we did this work nearly 15 years ago, where we focused on an antiviral protein called TRIM5 alpha. So we had known for a long time that rhesus macaques and other old world monkeys cannot get infected with HIV-1. Even if you were to inject HIV-1 into these animals, they actually cleanse out the infection. And it turns out that they do so at the intracellular level because their TRIM5 protein is able to recognize the incoming HIV-1 capsid to mount a very potent antiviral response. And that becomes a very effective way to actually purge the population of these viruses. It turns out we have a TRIM5 in our genome, but our TRIM5 protein does not have the avidity or the kinetics needed to interact with the incoming HIV-1 capsid. So taking a page out of this arms race sort of handbook, TRIM5 and capsids could be considered to be in this binding affinity arms race, where again, the interaction interfaces between these two proteins is being constantly chiseled away by rapid evolution. And indeed, when we looked at this nearly 600 amino acid protein, we found that most of the rapid evolution was actually concentrated in just one variable loop called the V1 loop, where 6 residues were very rapidly evolving. And this just shows the difference between these two TRIM5 proteins, where the human protein measured in the lab has only a 2-fold restriction of HIV-1, whereas the rhesus protein is a 100-fold restrictor and is remarkably good at defeating the virus. Now, what's really interesting is that we basically just swapped this V1 loop between these two otherwise different backbones and found that just this rapidly evolving loop was sufficient to confer most of the specificity needed onto the previously ineffective human TRIM5 protein to confer gain of function protection. And even just a single amino acid residue change at one of these critical positions was actually sufficient to do so. So this was an interesting way to switch the paradigm, because instead of focusing on the most conserved parts of the protein, we were now actually instead focused on the least conserved parts of the protein, because this is where the arms race was actually playing out. So this turns out to be a very useful paradigm. And so not only can we use this to actually identify domains that are important and residues that are important for sort of conferring protection against pathogenic viruses, but we wondered whether we can use the same signature in the laboratory to actually elicit even more potent versions, so-called super restrictors, which would be even more effective than the best natural versions against pathogenic viruses. In a way, we are actually jump-starting natural selection by taking a page out of this handbook. And indeed, this has worked to be really effective. This is yet another antiviral protein we worked on called MXA. And you can see that the second column here is the wild type version of human MXA, which is the best natural restrictor against influenza-like viruses. But just by mutating 5 amino acid positions that are rapidly evolving in, again, another unstructured loop domain, we are able to elicit versions in the laboratory that are 10 to 20-fold better than the most potent natural restrictors that we can get, which means that there is actually a lot of hidden potential within these rapidly evolving domains. And although people have previously ignored these rapidly evolving domains and residues, that's where most of the potential for increasing antiviral specificity might lie. I'll sort of close the science part of this talk by just sort of telling you that it's not just immunity that draws our attention back to the speed limit graph. Of course, we expect immunity and transposon defense to be represented here, but we also see lots of gene categories within this histogram in the right-hand side, where there are genes that have really no business belonging in the fast-evolving class because they are completely essential for function, including genes that are completely essential, for example, for cell division, mitochondrial function, reproduction, etc. So you could actually pretty much find your favorite cellular process, and you'd actually find a fast evolving residue within these. There's many cancer-related genes, including the BRCA1, BRCA2 genes that are also under positive selection, which means that we have to sort of expand our view from just looking at primates to many other model systems like we have done to provide a good paradigm for us to actually study what happens if we were to reverse the rapid evolution associated with these genes. And from these studies, which I won't describe today in the interest of time, the picture is emerging that even though we sort of think about the cell and the genome as this beautiful, Swiss watch-like mechanism where a lot of parts interact with each other perfectly, this is actually not untrue, but there's also a very hidden sort of series of prey-predator interactions going on within the genome, where these gears and knobs that make up the inner mechanisms of the cell are actually evolving much faster than you would have expected, given their essential functions. And only by actually combining the cellular and the evolutionary view do we get a really true picture of what are the natural selection pressures that shape these proteins. I'm going to actually, since in the interest of the conference, just give you a little bit of my own personal experience navigating an academic career with a lot of kind of important transitions where a lot of mentors actually really helped me navigate these. Perhaps the most important advice I can give you here is that mentors really matter. My own life experience in biology started as a chemical engineering student at IIT Bombay. And really, I had only taken one class in biology in my entire 4 years of college. And this was an extremely influential class taught by Professor K.K. Rao at IIT Bombay. And what I learned from him, because even though I was not his student or a student in his department even, is that he was extremely generous with his time and extremely generous with his encouragement, and allowed me to believe in the possibility that I could actually do a PhD in biology, even though really there was no reasonable reason to consider that possibility. So I'm really grateful for him for starting me on this path. I then went to the University of Rochester, where I did my PhD under Professor Eichbusch. And I learned very different things from him. What I learned from him is that it's actually possible for you to be an excellent scientist and have work-life balance. He basically went home every day to cook dinner for his family. It didn't matter what was going on in the lab. I also learned from him that it was actually not important. It was important to be successful and rigorous and disciplined, but also very important to not be a jerk, because those things were not necessarily correlated with each other. And then finally, as a postdoc, I joined Steve Henikoff's lab at the Fred Hutch. And really, for the first time, the relationship between the mentor and mentee became more of a relationship between a senior and a junior colleague. And that was extremely uplifting, because given that opportunity, many of us rose to the challenge and really took advantage of the fact that he always had our back. So I'm really grateful for these mentors. When I started my lab, I actually never worked on antiviral proteins, but really 50% of my research program is made about this. And this really started from a very influential collaboration that I've really had for the last 19 years with my colleague, Michael Emmerman. And one of the pieces of advice I'd give you is that even though you've come in with a very clear plan when you're starting your lab, it's really important to reach out to your community to see what other opportunities for collaborations might be there, because you never know when a very important arm of your science could be mediated simply by what the local environment actually provides. So not staying open to what the local environment provides would be a mistake and probably take away one of the biggest resources you can have. One piece of advice that I've sort of learned over the years, not necessarily as an overt piece of advice, but something that I've recognized, is that different people are good at mentoring about different things. So don't really think about one mentor who's going to provide all of the pieces of advice you need for your career and your science. Think of mentors as a committee and really incorporate mentors at different career stages as you go along, and particularly pick people who are deeply invested in your success. In that vein, it's very convenient to find mentors who are local in your department, and certainly, I cannot necessarily give you very practical advice like some of the people who are the organizers of this symposium about succeeding in terms of funding in the local Indian environment. But having geographically distant mentors can also give you perspective and challenge you to think differently about your science. So it's actually, one thing the pandemic has done is really told us how these geographical barriers are really not really very special. So you can really seek mentors who are not necessarily in the same department and even the same floor. One of my most valuable mentors in the last 19 years has been Sue Biggins, who's actually currently my boss, but for many years was just the junior faculty, next most junior faculty member. And everything that I was actually going through, she had just gone through 2 or 3 years before. So although on the one hand, it was really useful to actually have mentors who are very senior, actually the most practical advice you often get are from people who are either at your career stage or just one career stage ahead of you. So do not ignore the possibility that in terms of actually succeeding, they have already figured out some of the mechanisms they need, and you should take advantage of that. I will not actually tell you that luck does not play a role. The whole point is that we know that luck can play a pretty significant role, and small differences can make a big difference in terms of ultimate success for the lab or grant funding, etc. So the goal is to seek good mentors who will invest time to make sure that you avoid making the mistakes that will actually potentially harm or hinder your career. So that's where the practicality comes in. As you sort of transition to running your lab, etc., it's actually really important to consider the possibility that whatever you invest in your people will actually pay back for you. So hire awesome people. Be unafraid of ending up in your trainee shadow. I have trained many people who are far more famous and successful than me, and that's actually a really good thing. It's been a good thing for them, and it's been a good thing for me. And even in fields, it's really important to recognize that science is not a zero-sum game. And there are many fields that can actually basically end up killing themselves because they're extremely hostile to newcomers and to new investigators. And so while healthy competition is a really good thing, unwelcoming fields will eventually run out of really good people because people will go to other fields, since talent will be able to seek out where the opportunities are. Just to again reiterate, your trainees will be your biggest legacy unless you're going to actually end up winning a Nobel Prize, in which case I'll probably accept an exception. So it's really important for you to treat your trainees well, because ultimately they will go on and propagate your way of thinking in science. Finally, as you get more and more senior, and actually by being senior, I don't even mean a senior professor, even being a professor has privilege. And I hope you will recognize all the people who use their privilege to help you, and you will use that same privilege to help others when you get that opportunity. And it seems unbelievable that you have that opportunity if you're just looking for your first faculty position, but actually there is definitely somebody who's looking up to you, and you're in the position that they would love to be. So definitely consider the possibility of paying it forward. And finally, we pay a lot of attention to the people who are doing the shiny stuff, who reach the national academies and get the papers in the Cell Nature Science journals, which is all very fantastic. I refer to those people as the tiles because they're really attractive and they basically attract you to join a department or join a particular lab. But the people who actually make it work, for example, including the people who are the organizers of the symposium that we are all attending, those are the people that actually make it worth being in science and really sort of make it hospitable and welcoming. So I hope you'll take some time to actually thank the people in your local sphere that make it welcoming for you. For me, these three senior scientists who've been part of my lab since I've started, Danielle for the first 5 years, and Ida and Janet since, are really the sort of lifelong blood of the lab that I'm really grateful for enduring and ensuring the success of the lab. Thank you very much.
Thanks very much, Harmit, for that excellent talk, both on the scientific front as well as very valuable lessons for someone at various different career stages. We'll take questions if there are any. I can see one question on the chat box. We'll start with that. So, do you think that directed evolution is what is happening in the context of COVID-19 epidemic?
Yeah, I don't necessarily think of it as directed, except that it's navigated by where the opportunities that natural selection sees. So directed evolution is a loaded term in evolutionary biology because people sort of tend to think about it as a somewhat Lamarckian aspect of it. And there's not a Lamarckian aspect here. It is basically as we are battling and we are developing antibodies, either because of infections or because of vaccination, the virus is basically finding remarkably new strategies to escape that. Omicron is a very perfect example. Whereas Delta was like what we would consider like a normal kind of evolutionary step that would be taken as most people were encountering the the virus and then getting vaccinated. Delta was not really mediated by a vaccination sort of strategy, but almost certainly Omicron was mediated. So we are basically intervening by the course of our vaccination strategy to really dictate the course of evolution for the virus. And probably that'll actually continue because we obviously can't stop boosting people, hoping that that'll actually prevent the virus evolution to happen. And so we expect many more jumps. And really what makes it all possible is the fact that the effective population size of the host for these viruses is incredibly large. And there are pockets of the world that are subject to different levels of vaccination. So unfortunately, because of that, there's unequal selective pressures. And so the virus can really thrive in certain pockets and then make it back into the vaccinated world.
All right, thank you. This is a question on the second part of the talk. Any advice on how to identify the right collaborators and trainees, especially in the first few years?
Yeah, that's a really great question. I do actually have some practical advice. So the advice I give to junior faculty who join my department is that you need to devote a couple of afternoons a week, every week, to meet with people within your department and within your local community, have coffee or have lunch with the people there. Because, the whole process of getting selected for a job, especially in a pandemic, means that you actually have very little face time with the people who are going to be your future colleagues. So it's really important that when you show up, which is often like a year after you've actually interviewed, you reestablish those connections and see where are the opportunities to actually have that. Having said that, you cannot actually sustain collaborations with more than 2 or 3 people. So part of this job is actually to be really kind of brutal and selfish in terms of where there are opportunities where there's actually an equal collaboration rather than sort of this unequal collaboration where you're actually ending up doing service work for some other big established lab. That's not a good collaboration. I mean, you might want to do that because you get some sort of funding in return, but that's not what I mean about a collaboration. A true collaboration is where there's intellectual contributions made by both labs that are somewhat on equal footing. A perfect example is what I have with my colleague, because my colleague is the virologist. I'm not the virologist, but I also bring in a perspective that he does not have. And those examples are actually not as rare as you might suspect. All of you actually have training and specialization in something that other people will find valuable. It's your job to basically look broadly, but actually then establish connections. I also have a 1-strike rule. It's not a 3-strike rule, it's a 1-strike rule. Whereas if a collaboration in the nascent stage shows any sign of selfishness, that's it, because life is short and you really need to protect yourself as well as your people, especially when you're in a young lab.
Excellent advice about that 1-strike rule. All right. there any practical tips to spot a good mentor?
Well, I wish I could tell you that. Certainly being lucky helps, but obviously that's not going to be useful. I will just say that, you probably want to pick people who have, I think you can actually look at what, for people who are established, you can actually look at their career path and their trainees. I certainly think that it's actually worthwhile to look at that anyway if you're thinking of joining a lab or a department. However, the young people fared in the department, how did the postdocs from that lab or graduate students from that lab do? That's always good advice. And conversely, you should recognize that people are looking at that for your lab as well. And so definitely treat your people well, because people will reach out to them. It's always a little suspicious to me when I look up a lab and there's really no links to previous alumni, even though you go, well, you probably are in touch with these alumni. And so you always wonder, like, what is the PI trying to hide, etc. So it's really good to be completely transparent. Please do reach out to all of my current and former students and find out whether this would be a good advice for you. Really, honestly, nobody else can give you as honest advice about mentoring as people who are in that environment or have been in that environment recently. In terms of mentors, I think what you have to pay attention to, how much attention do you pay? So a good example is that we have a mentoring committee that's mandatory at my institute. I serve on the mentoring committee of several junior faculty right now. And some people take that very seriously, which means if you were to give me a grant application, you can be assured that when you get it back, it'll be full of red, right? Because I'll have actually edited it as if it was actually my own paper. And now you might not want that kind of attention, but that's where I feel like I can give you my best, advice. Other people will just look at it and say, it looks fine. And I mean, it's not that they haven't read the paper, but they haven't actually given you the fine-grained investment and advice that's needed. That's not to say that they won't be good mentors for different things, but they would definitely not be good mentors for the writing of and success of grants and papers. So that's what I mean. Different people provide mentoring for different things. You might actually need just the right epigenetic imprint in one way, and even just one or two interactions would be sufficient. Whereas there are going to be these long-term relationships with mentors where you go back again and again to them because you know that they have your best interest at heart and they're really invested in that. That investment is where you find out whether this is going to be a really good, productive relationship or not.
Thank you very much, Harmit. I think those were excellent points. There are other questions in the chat box, but in the interest of time, we would have to move ahead. So thank you once again, Harmit, for the exciting scientific,
thank you,
as well as very valuable advice to the entire gathering here. Thank you so much.